All right, let me keep going. The second speaker is Dr. Dave Edwards, and the title of his talk is Improving Genome Assemblies and Capturing Genome Variation Data for Applied Crop Improvement.
Well, thank you very much. It's really good to be back here again. I'd like to especially thank Rajiv for inviting me to this meeting again. It's interesting to see the number of years that this has been going on now, and to see the increase in size and a number of researchers that are coming here. It's a credit to Rajiv that he's been able to grow it so successfully. So thank you very much for that. I've talked previously about some of the work I've been doing in a broader sense at previous talks here. But in this talk I thought I'd change it a little bit. So this talk's going to be a little bit different, especially to the last talk, in that I want to focus down a little bit more on one area, which I think would be quite nice to concentrate on. And the reason for doing this is that some of the work I've been doing on trait association, on hand genomics, and on looking at structural variation has been hindered by the lack of high quality genomes. Now, as everybody knows, genome sequencing has expanded hugely. The cost of sequencing has dropped massively. And one of the consequences of that is that there's been a general decline in genome assembly quality. And many of the genomes that are assembled now, even the better quality ones, still have limitations. So over the last few years, what I've been doing is working on methods to both assess genome quality and also to then take that information to try to improve the quality of some genomes that are out there. And I'm going to give some examples in this presentation today of what we've been doing that in a few different crops. So I'm going to talk a little bit first about our work on wheat, which has been going on for a while, some chickpea work that we've been doing with Rajiv. I'm going to talk about Skim GBS-based genome assembly and validation. Now, Skim GBS is different to the GBS that you're probably familiar with, so I'll talk a little bit more about some of the details of how that works. It's quite a different approach. It's got different aims. It's more expensive in some ways, but it's very targeted, doing some things particularly well, and I'll describe how we've been using that. And lastly, I'll talk a little bit about the chronology now, which is probably some of our most advanced work because we've been working at that for the longest, and we have quite a bit of funding in that area, but a lot of this stuff I can't really talk about because it's commercial, but now that some of it's been published, I can talk about that. So starting off with wheat, I was at a PAG meeting back in, I think it was 2010, and I'd just given a presentation on some work we were doing with NGS sequencing and canola at the time, which is very early on. If you think back at the technology then, it was still reasonably basic compared to what it is today. And I was approached by someone by the name of Jaroslav Doricel from the Czech Republic, and he came and asked me whether it would be feasible to sequence wheat chromosome arms using this next generation shotgun approach. And at first I thought it was crazy. How are you going to get isolated wheat chromosome arms? But then he explained that he could do this. He could flow sort them and get microgram quantities of these. So we had a bit of discussion. We thought it would be worthwhile trying. We realized that it was probably worthwhile trying just from some amplified material. So rather than spending weeks and weeks and weeks to generate large amounts of material, just spend an afternoon, isolate some chromosome arms and amplify them. And we went through and we did an assessment of the data, and we did an assembly of that. And that resulted finally in a publication back in 2011 in the Plant Biotechnology Journal. We got the front page. And it was quite difficult to get this work published because people were quite skeptical of the ability to go ahead and sequence and get a reasonable representation of the gene content from a combination of next-gen sequencing was one, which still hadn't been proven hugely, and also the isolation of chromosome arms, which people thought were quite messy. And there's still a debate going on at the time as to whether people should be using things like 454 sequencing for this kind of work or the Illumina. But we managed to demonstrate that we'd assembled all, if not nearly all, of the wheat genes on this chromosome arm, and we managed to put more than 50% of them into a synthetic build, and we released all the data and put it on the web. So that was my first foray into wheat genome sequencing. And because we were pretty happy with the results, we carried on and did a little bit more. We then went on to sequence the chromosome arm 7BS, and we managed to show that there was a translocation between 7BS and 4AL We managed to, the translocation had been shown previously genetically, but we managed to show that translocation and narrow it down to 3 or 4 genes in terms of distance. These genes were absent from both regions. We managed to really get fine resolution mapping of this translocation. Finally, we completed all of the group 7 arms, long and short, so for 7A, B, and D. We did quite a lot of analysis of this. We looked at gene loss. We did some network analysis of gene loss, and we managed to show there were signs of domestication through following the tetraploidy event. Following the diploidy event and with the coming of the tetraploid, making the hexaploid after that. So we managed to show signs through this network analysis of lost genes of the domestication and dispersion of the breadwheat genome by the early farmers, which is quite nice looking back using the latest next generation sequencing technology and translate it to what people were doing in fields 8,000 to 10,000 years ago. So we're quite happy with that. Then the International Consortium had decided to adopt this approach, and I'm an author on this paper as well, but I'm a relatively minor author. But they then followed that approach to sequence all of the chromosome arms of wheat, and this came out in Science earlier last year. But there was a bit of discussions and some rumours going on that there were some issues with the assembly. So when we got hold of this, we decided to look at it in a little bit more detail to try and find out and compare it with the assemblies that we had before and to see if it could be improved. One of the first things that we looked at is Contig duplication. Now we're classifying this as an arbitrary measure of regions which are 1,000 bases or more, which are represented more than once within a chromosome arm assembly. Now everybody knows that wheat is repetitive, so if you had a full representation of that arm, you would expect to see many of these repetitive regions. However, you've got to discriminate between the true repetitive nature of the wheat genome and the duplication, which is an artifact caused by the parallelized assembler that they use, which it's renowned for. And given the data that was used for this, which was paired end Illumina data, it was relatively short. There should not be information which allows you to develop contigs or duplicate contigs of greater than 1 kb. So if we're seeing these things, they should really be artifacts of the data caused by the assembler. So what we're seeing here is some chromosome arms in particular are having a very high proportion of duplicated regions, and these should not be reflecting the biological replication of the wheat genome, but actually a fault in the assembly process. So if you're actually using this data for any kind of structural analysis, it's going to cause loads and loads of problems, so it's something to be aware of. Another thing that we were surprised about is that there are a certain number of genes which are annotated in the assembly, and when you went back to the raw data that was used for the assembly, there were no raw reads supporting their assembly, supporting these genes. So I've got no idea how these got in. I've spoken to a few people. No one else can give me an explanation for this. But we can be fairly confident that these genes are not actually on these chromosome arms by the fact that there are no reads from those chromosome arms in the assembly. If anyone's got an idea as to why this is happening, I'd be very happy to listen. So we went away and we did a reassembly of this data. We do not see any duplication in this new data, so we think it's fairly clean. We didn't use a parallelized assembler. We used lots of memory, but fortunately we've got access to 5 1 terabyte memory machines, and we've recently got access to a 6 terabyte memory machine, so we had the capacity to go ahead and do this. In some cases, our chromosome arm assemblies are longer than the published IWGSE ones. In some cases, they're shorter, but we're not seeing the artifact duplication in there, and we're also not finding genes which are not represented. So we haven't published this, but we are using it for further characterization of the genome. We're making synthetic builds from it. We're using Skim GBS data to make representative pseudomolecules from it, and we're also using it for SNP discovery. From the group 7 chromosomes we published last year, there were 4 million SNPs we identified there. These are homolog-specific SNPs. These can be assayed in a homolog-specific manner. So I'm not saying there's just 4 million SNPs in the genome. There are a lot more, but there are 4 million SNPs across group 7 that we can assay in a homolog-specific manner, and these are really useful for genome validation and for trait association. Across the whole genome now, we have more than 20 million SNPs for wheat. And again, these are homolog specific and can be assayed in a homolog specific manner. So we think this is a really good resource. Moving on to Chickpea, there's been quite a few people here working on Chickpea, including Rajiv, and we worked with Rajiv on the draft genome of the Kabuli type. And then, as many of you know, there was the Desi type was published shortly after in the Plant Journal. So we were interested when this came out to have a look at the two, because from what's known about breeding and evolution, that they should actually be looking very similar. And when we had to look and compare these two genomes, they were not quite as similar as we expected, and particularly even on a whole chromosome level, we're seeing some similarities between chromosome 6 and chromosome 2, for example, which shouldn't really be there, and chromosome 8 and chromosome 3. So we thought, well, what can we do to have a look and see if we can validate this assembly and find out which one's right and try to improve it. So following the approach that we'd worked with in wheat, we went to Yaroslav and we asked him, is it possible to isolate individual chromosomes from Chickpea? And he said he could do it for most of them. There's 2 chromosomes he couldn't separate, but he could provide those as a pool. So we did that. We did that with the Kabuli genotype that was sequenced. So we're mapping exactly the same genotype against that was used to generate the reference. And what we see is general concordance, showing that the Kabuli reference is in fact very good, and there are no major errors. There are about 40 regions, which are quite small, which do appear to be misplaced on the assembly and don't belong on those specific chromosomes, and they belong elsewhere. I think you'll see in most genomes you'll actually see more than this type of error, but this gives you an idea for a particularly good reference genome that there are still some things wrong that you can go through and identify. And one of the advantages of this is it's sequence-based. You can get down to the base pair resolution of saying this Contig here has been misplaced. It needs to go onto a different chromosome, and we know which chromosome it should go onto. We're now applying this approach to a whole range of different species. It's really quite useful, and Yaroslav's even developed further methods to extend this. So we took the data and then mapped it to the, we generated Desi data from the same chromosomes and compared the mapping of the Desi data and the Kabuli data to the Kabuli reference. And one of the questions we were asking here is are we seeing a difference? If we're taking the Desi chromosome arm and mapping it to the Kabuli, if there was a major difference between the Kabuli and the Desi, you would see differences in patterns. What we're seeing is pretty much exactly the same pattern. There's some very minor differences, but pretty much exactly the same pattern when you map the Desi and the Kabuli chromosome arm data against the Desi reference. And this is telling us that, at least for these chromosomes, at least for chromosomes 3, 8 and 5, that Kabuli and Desi, there's not a huge amount of difference between them. So what's happening with the Desi reference? So we took the chromosome sequence data for Desi and Kabuli, and we mapped them, and we could see quite clearly that this is where the problem lay. In that, so if you have a look at chromosome 3 down here, this is the chromosome 3 from the Desi reference. If you map reads from chromosome 3 for both Desi and Kabuli, they mapped a part of chromosome 3, but only part of it. This region here, which is part of the Desi chromosome 3 reference, it actually had reads mapping from chromosome 8. So what they've done, they've effectively merged in generating this reference, chromosome 3 and chromosome 8, and there are a few other errors that were picked up around here. So we'll start to pick up some major issues with this. One of the things that surprised us a little bit was these purple bits. When we get the chromosome arms, they're not pure. There's between 10-20% contamination with other chromosomes, and that's why you see this sort of shading, where you're seeing here, for example, mostly chromosome 5, but you're seeing other hybridization or the mapping of other reads to other parts. But these regions, we're not getting any reads mapping to them from the isolated chromosome arm material. So we got some whole genome assembly data from Desi, and again, this is the exact Desi variety that was published and used to produce a reference. And again, we're not getting reads mapping here. When we look at these regions, we've got no idea what they are. They don't appear to be Chickpea. There are no genes in there. There are no transposons. This just seems to be some random ACs, Gs and Ts. We've got no idea, even at very low resolution, what these are. We published this recently, published this last year, I think, in the Plant Biotechnology Journal. So if you want to read that about some more, that's in there. So identifying errors in assemblies is one thing. It's easy to point at things that are wrong once you've got this sort of information. But how do we improve it? What mechanisms, what tools have we got to go through and improve assemblies? And there's discussion earlier about the standard GBS approach using metal restriction digestion to develop lots of markers to be able to go through and generate genome assemblies. And that approach is really good, and it's particularly cost effective, it's quite cheap, but I would argue that it's not going to get the full resolution of markers that you would get with this method, which is the Skim GBS method. So I'm going to talk and expand on how we do this. So with Skim GBS, we determine it's a two-step process. First of all, we identify the SNPs. So we sequence the parents, we use our own tool called SGS Order SNP. This is published, it's freely available, it's used for a whole range of other projects. We identify lots and lots of SNPs, and this is what we use to identify the 20 million SNPs in wheat. Once we've got that, we do low coverage skim sequencing of the segregating population. There's no restriction enzymes, we just go through, generate some data. It's not selected for at all. We then map these reads to the reference genome, and where a read from the population covers maps to a previously defined SNP, and we have to be fairly accurate with our read mapping parameters, we can then call that SNP, and then we can impute and clean to define the haplotype status. So graphically it looks like this. This is part of the genome. We initially identify SNP polymorphisms across the different contigs and references, and then with the population reads, if this read says it's an A here, we'll then just call that an A. Ideally, this is what the haplotype blocks look like. You've got the parents, and then you see in most regions there's no recombination. Regions of recombination should look like this. That's a very pretty cartoon. It would be really nice if it looked like that. Unfortunately, it looks more like this. This is pre-imputation, and you can see there's a reasonable amount of missing data here, but we also have quite a bit of resolution as well. We can do some fairly basic imputation on this, and what you start to see is these blocks here, which are clearly misplaced. The haplotype segregation, these are SNPs going along the top. These are individuals segregating going down the side, and we can see within this region of the genome and these other regions of the genome, these pieces of the reference have been misplaced. They do not belong here. So we can actually cut these out and then rebuild the assemblies. But not only that, the pattern that we have going down through the population acts as a barcode. We can look at that barcode and we can ask whereabouts in the genome assembly do we see this pattern, and then we can start to place these contigs in their correct position. And we've been doing that for quite a while in canola, and now we've been applying it to a range of other species. And we've recently got an improved Kabuli assembly based on it. It's slightly larger than the other one, but not very much. And we don't have the issues of the rearranged blocks and the misplaced blocks on different chromosomes. The Desi genome is vastly improved in comparison to the one that was published. And again, we have these are the initial, these are in scale and size. These are initial published pseudomolecules, and these are the ones that were produced recently. And as you can see, they're a lot cleaner, and we think they're much better references. They're not perfect, but they are a lot better than they were. This is just a graphic showing the improvements over the initial Kabuli to the latest version of Kabuli that we've got. It's not huge, but the improvement from the original Desi to the current Desi is a vast improvement. We still think there are things we can do here, but we think for now this is working pretty well. When you look at the comparison between the doproptin, Kabuli and Desi, we are seeing some regions which are not aligning very well. We're not quite sure what's happening here. It could be that there are true differences between Kabuli and Desi at these regions. It could be an issue with the mapping population that we've used in terms of recombination at these regions. These are the regions that we need to look at in more detail to try to validate it. So moving on to canola. Been working on canola for probably the longest. We initially approached, I think back in 2007 by Bayer, asking if we'd work with them on assembling the canola genome. We had a press release in 2009 showing that we'd assemble the first draft assembly. That assembly was used in 2009 to identify shadowproof genes and to characterize them, and that technology is now being used, and varieties that are now being released by Bayer, which show increased or reduced shattering, which is an important trait. That trait you can actually trace back to the use of the genome sequence early on. So even from the very start, the genome assembly has been used for trait improvement, which is quite nice, and there are quite a few other traits that have developed from that. But that first genome it's had quite a bit of extra work, and we've been expanding with lots of other genome projects, and this is a public canola project which I'm a co-author on, which was published only a few months ago. But when we looked at that genome, the reference was produced by Genoscope, and they sent us a version, and we said, well, we will try to improve it before it's published, but they said, no, it's too late, we've already started doing the annotation, we have to work with this. But we knew it wasn't perfect because we had proprietary assemblies that we could compare it with. So we went in and just using some public data we tried to improve it, again using the Skim GBS. So we had 92 double haploid individuals in this population, and then we did Skim sequencing of 1.68x coverage average of that. We used an algorithm which we've used for Chickpea and a few other species called Contig placer. And this is an algorithm which essentially takes us these barcodes and allows us to put the contigs back into the correct position. And there's a discussion earlier about when you've got too many SNPs, what do you do if you've got 20 million SNPs? You're not going to use all of them for genetic mapping or for any sort of analysis. What we do, we develop tagging SNPs. So we identify SNPs which represent haplotypes in particular groups. So we can actually shrink the data set there, and that makes the maths and the computing a lot easier. So with this data, we managed to identify contigs which had been misplaced. We managed to place contigs which hadn't been placed. We identified chimeric contigs which could then be split, and been using that to improve the assembly. So we had just over 1 million SNPs in Chickpea between these varieties. 60% of the alleles were called, and then after imputation, we had about 90% of the alleles were called. And then we took this data into the Contig placer. Before Contig placer, this is the size of the genome, about that 75%, so almost 76% of it had been placed, and then afterwards we managed to get 93.9% placed within the genome. Now, interestingly, the genome assembly, I thought I should slide on this. Okay, so going back to here, I think I skipped over this. The genome is it's a polyploid. It's got the A and the C genomes in there. The A genome has 10 chromosomes. There are 9 chromosomes of the C genome. When we got the first reference, there were 22 sets of unplaced contigs, and these consisted of contigs which could be placed onto a particular A or C chromosome, so there are 19 sets of those. There were contigs which could be placed only on the A genome somewhere, but we didn't know where, or only on the C genome, and none of these could actually be placed physically, we just knew that they were around there, and then one set which was completely unplaced. And when we did our analysis, 98.5% of the unplaced contigs which had an initial chromosome assignment mapped to the same chromosome, but in our case we can actually physically place them on that chromosome. So we improved the assembly substantially. There's just under 94% of the assembly has been placed into pseudomolecules, and we're very confident that these are placed correctly. The remaining unplaced ones, they're relatively small contigs, and there are either no or not enough SNPs on there to be able to place them accurately. We require at least 2 SNPs as a measure of confidence to be able to place them. And some of the contigs were quite messy and chimeric, and we haven't got around to splitting them up and replacing them yet. So even with relatively finished genomes, you can actually go through and use this data to improve them, to validate them, and to have a lot more confidence that they're there. There's another paper that's just been accepted in TAG. It should come out fairly soon, and this is when we were looking at the recombination across the genome and also gene conversion. There have been a few papers that were suggesting that gene conversion in Arabidopsis, for example, was very high. And when we first started looking at our data and using the same parameters that they were using to call gene conversions, we were seeing very high levels of gene conversion. But we looked at it more closely, and what we saw was that these are not actually gene conversions. These are minor structural errors in the assembly, which look like gene conversions. So we managed to go in there and managed to resolve some of those and put in some extra parameters. And when we did that, we got a more clear view of the level of gene conversion. And again, if you really need to have this sort of density of SNPs segregating in a population to be able to measure gene conversion to this sort of accuracy. So again, I think this is a useful method. I think across many genomes there are still going to be some minor structural structural errors in the assembly that need to be fixed, but we're kind of getting there in starting to resolve these. And this, as you can imagine, is really crucial. If you're doing fine trait mapping, if you want to get down to a particular candidate gene and you've got all these markers in the area, it doesn't matter how many markers you've got, you really need to have your reference genome to be reasonably accurate in that area, because otherwise it's just going to throw you off. So I'm going to conclude here. I think I've got a little bit of time for questions. And the conclusion is that many high-quality published genomes, and I'm not being critical of these genomes, I think the genomes that are published are fantastic, and at some point you've got to draw a line and publish them. I think people are doing that and publishing them at the right time, but they can still be improved, and as the technology improves, we are seeing much better genome assemblies. And that's quite clear when we looked at the gene conversion amounts. I just did the rough estimation at the beginning. We saw very high gene conversion in the A genome of Brassica, less on the C genome of Brassica diploid that was published, and even less on the Chickpea genome. And that's a factor of genome complexity and the timing of when it was done. The A genome was published first, I think back in 2011 or 2012. The C genome was published far more recently. The data was a lot better, the algorithms were better, and then the Chickpea was, it's not a polyploid, so it's a bit simpler, and again, it's a very recent genome, so the quality of that was better. So you can actually really have a look at the quality, use this as a metric to establish the quality of reference genomes. Chromosomal genomics, Skim genotyping by sequencing and bioinformatics can validate and improve genome assemblies. And I think this is going to be really crucial to go through and look at some of the genomes that have been assembled out there and any new genome projects that are coming up. So really consider whether these approaches could be used in addition to all the PacBio and normal GBS type approaches to complement those approaches to improve the assembly. I think quality genome assemblies, they do improve trait association. We've been doing quite a lot of trait association with these genes, with these genomes. And once you've got all the SNPs, you will find SNPs which you think should be associated in that region, but they're out, and really it's just because that SNP's in a misplaced Contig, and you've got to start sorting all that out. The quality genomes are essential for pan-genome assembly. So one of the things I've been looking at in terms of pan-genome assembly, to be able to look at presence-absence variation across genomes, we have to differentiate between true presence-absence variation, which is biological presence-absence variation, and presence-absence variation, which is due to differences in the assemblies. So if you compare our wheat genome assemblies with the published IDW GSC wheat genome assemblies, you might say there's presence-absence variation there. It's exactly the same data. It's just a difference in the assembly, and I think that's crucial to be able to differentiate those. And I think something that's going to be more important down the track, and especially as the technology for long-range sequencing is coming through, the improvements of the PacBio sequence data, for example, the BioNano, I think, is particularly useful, and just the ability to produce really nice mate pair libraries from Illumina now is really helping us to look at large-scale structural variation, which we couldn't look at accurately before, and I think that's going to tell us some interesting things about crop genome evolution. So finally, I'd like to thank the people who did this work. I don't do any work, as people know me. I tend to wander around conferences giving presentations, but I do have a fantastic team. We've now moved to the University of Western Australia, which is a beautiful place. If anyone happens to be in that part of the world, I welcome you there, and I'd love to show you around. But these are the team who, in my group, who've done most of the work that are presented here. There's people who have left the group, who have contributed significantly to the work that are presented. Jackie's group, we work very closely with Jackie, and Jackie's in the audience, and is my main collaborator as well as wife, as you know. The colleagues in the Czech Republic, and also colleagues on some of the Chickpea work, again, who are here. And the various people who have funded us, the Australian government. I'm now at the University of Western Australia, but I still have an adjunct position at the University of Queensland. Illumina are always very supportive. The AGRF have always been very good at getting the sequence data. QSIF helping us manage the data resources. We've got a huge amount of data infrastructure and computing infrastructure behind us, and ICRISAT as well through various collaborations. So with that, I'm happy to take any questions. Thank you.
Okay, let's see. There we go. So, in these various publications or somewhere else, do you have a nice outline of basically these are the basic sanity checks you do on a whole genome in order to make sure it's okay? Like basically, is there a place we can look for to see what the list of all these things are?
That's a good question. I was actually thinking about that over the last few days, and when I was thinking of preparing this presentation, I realized there isn't anything out there that's like that. There are a whole range of things like looking for the number of conserved eukaryotic genes, which are standard measures in there. It's very difficult to reference because you don't always get all the genes in the assembly, they're not always in your genome. But you're right, I think we could do with something like that, and I think Nature Genetics would be a good place to put that. Yeah. So I think expect an expression of interest or a draft coming through. I think that'd be really useful, and maybe we should talk about that as maybe something to work on. I think it'll be useful.
One more question? All right. Thank you.
Thank you.
So our next speaker is Doug Cook to talk to us about